in terms of defining stem cells. Nanoparticles, which are either derived from cells or actually artificially produced as pharmaceuticals. And then, of course, nucleic acids like DNA, double helix of DNA, or RNA that comes from DNA. And, of course, biomaterials that act as inductive scaffolds or guides for cells to actually engineer new organs. So that's sort of the toolbox of regenerative medicine. Next slide. Just wanted to remind everybody what RNA is, because I'm going to talk a little bit more about it and why it's significant. Ribonucleic acids. The ribonucleic acids, so the way they are derived is from DNA. So everybody has a sequence of DNA, and we can all be sequenced individually. That sequence of DNA determines what genes can be expressed. And by expressing a gene, that means we make messenger RNA that can be then, so we transcribe DNA to messenger RNA, and that messenger RNA can be translated into a protein. So that messenger RNA sequence determines the amino acid sequence of a protein. So we go from DNA to RNA to protein. But messenger RNA that makes proteins, that codes for proteins, is not the only RNA that's made from the DNA template. The areas that we used to call junk, now you should all know now, if somebody says something's junk, it's not junk, right? The stuff that we used to call junk that that was in the DNA code was located in an area called introns. We call it junk. We now know makes a whole bunch of different non-coding RNAs. That means that these are never translated into proteins. So the idea is, so what are all these tiny little RNAs doing? Well, it turns out they're not junk, and I would never call them junk to their face, because they control, next slide, they control all of these messenger RNAs. That means that what they do is they feed back and they say, okay, you made a messenger RNA. You are not becoming a protein. And they can do that, they usually do that by silencing, by actually binding to this messenger RNA and cutting it up. They can also just hang on to it and say, okay, for a little while, I'm going to hang on to you. You know, like one of those illegal holds in hockey. I'm going to hold on to you for a little while, and then I'm going to let you go. And that usually means that they have core binding, but that means that they can go and silence somebody else for a little while. So these microRNAs, they do nothing to change the gene sequence. So they're not changing the genetic code. They're not having a genetic effect on the organism. They're affecting the gene after it's been already turned into messenger RNA. So they're having what's called an epigenetic effect. So they affect after the gene has been actually transcribed. And so, but they're extremely powerful. In fact, the location of that junk happens to be in the DNA code right next to the stuff it's silencing. So if you take a whole gene out, you'll see, okay, well, there's this length of DNA that codes for the messenger RNA and the protein. Right next to that is a junk piece that codes for silencing it. It's like an extended family. You know, it's like people agree and disagree all the time, right, in the family. So they're just always feeding back on each other. So that's the silence. And why are we so interested in this? Because this is highly conserved all through the animal kingdom. You know, there's 2,000 or 3,000, maybe up to 4,000, depending on what species, of microRNAs that are known. But, you know, better than 40,000 different sequences of messenger RNA. So there's a smaller number of sequences here, but they can also block a lot of different messenger RNAs. So they're very powerful, and we can make these in the lab and use them as treatments. So we can actually make silencing microRNAs. So epigenetics does not change the gene sequence. It alters the relationship between the gene and environment and their interaction. So identical twins with different hair color, epigenetically determined. The dog with two different eye color, that's epigenetically determined, not genetically determined, for examples. Most diseases, spontaneous diseases of people, like heart disease, diabetes, are epigenetically controlled. There are many genetic diseases, as we know. There are many genetic predispositions. But the course of disease is largely determined by the epigenetic control of those genes. Next slide.